Hi everyone, my name is Shmari and I'm one of the tutors at HD Academy, and I will be teaching you a little bit of biology today, get you excited about bio. Alright, so first I'm going to draw a cell. Sorry, I'm not very tech savvy, so let's see how we go. There's my cell right there. Now I'm going to draw in the organelles, which are like the organs of the cell. So let's try some new colours. We'll get green for chloroplast. As you know, this is where photosynthesis occurs, and it sort of looks like this in shape. Okay, next I'm going to pick a new colour, probably red, for our mitochondria. Now these organelles do the opposite of chloroplast, so they break down glucose, and they sort of have this kind of cristae-like structure. There we go. Now I'm going to draw in the nucleus, which I'll do in blue. Okay, so the nucleus is quite large, and it's also got a dark little patch in here called a nucleolus. There we go. And I might also draw in some rough endoplasmic reticulum in, how about we go with orange? It's a nice colourful cell for us to work with. There we go. And because it's rough, it's got the ribosomes on the outside, because we'll be working with these a bit later. There we go. Now, what we're going to be talking about today is protein synthesis. Now, if you're at home watching this, if you have time, I'd like you to think of the two main stages that occur in protein synthesis. They both start with T. Alright, so one, hopefully you came up with transcription. And number two is translation. So first I'm going to start with transcription, which occurs inside the nucleus. But before I do that, I'm going to change the colour to black. Okay, so now we're going to be working in this area, inside the nucleus, because transcription occurs in the nucleus. So as you may have learnt at school, the nucleus is the storage area for DNA. And DNA has a double helix structure, kind of like a ladder, that we can look at over here. Now, if I take that and make it larger, you'll be able to see the bonding that forms between bases. So the four bases are adenosine, thymine, cytosine, and guanine. There we go. Draw those in. So when we're doing transcription, we don't really want all of the DNA to be copied using these nucleotide bases, because that would get really confusing, wouldn't it? If we had all this DNA being copied constantly and moving out of the cell and moving in, there's not really a system. So to combat this, we have mRNA. So I'm just going to change my colour so we don't get confused to draw in our mRNA. And I will go with, how about light blue? Okay, there we go. So basically what happens is, in our first stage of transcription, an enzyme comes in. This enzyme is called DNA helicase. And what DNA helicase does is it comes in and it unzips this double helix pair so that we can gain access to our instruction sheet. So there we go. It's unzipped it for us nicely there. Now it's mRNA's turn to shine. So I am going to go with green. There we go. And what happens is, so we've got our exposed nucleotides here from our DNA. And just to stop us from getting confused, I'll pick pink here. Okay. And it pairs up. But the thing is, mRNA differs from DNA in the way that RNA has different bases. So instead of thymine, we've got uracil. It's got uracil, adenosine, as was the same with DNA, guanine, and cytosine. So the pairing occurs in the same way. So instead of T, we have a U here, G, and can you guess? A. Exactly. So this is our mRNA, and this is what we'll be focusing on next. So basically, this mRNA has copied the sequence on the DNA, and this is all occurring inside the nucleus. This tiny little nucleus, all these big processes occurring. Now that we've copied our DNA, we can move it out because we need to use our set of instructions. So this moves out. So you're probably wondering now, what happens next? We've copied it, so what? Like, why are we doing this for? Well, the whole purpose is now to get these instructions across so that we can synthesize some protein. And proteins are essential for everything. Basically, our skin, our muscles, everything requires protein. It gives us energy. It creates our enzymes, which speed up our reactions. They are amazing. So this process is essential. Okay, so now we've had step 1 here, transcription. And now we're moving into step 2, which is our translation, which occurs in the cytoplasm. So we're in the cytoplasm now. So in the cytoplasm, there's these little things called tRNA, which kind of look like little T's. They're cloverleaf shaped. And what they're able to do is they're able to read our mRNA that we just drew here. So mRNA, if I draw one over here, is really long. So can you imagine that? Times that by like 1,000, and it's still going to be way smaller than what a real mRNA. It's close to 100,000 nucleotides in length. So that's a lot to read in one shot. So instead, what happens is the mRNA is read in groups of 3, which are called triplet codons. So this is where our tRNA steps in. So our mRNA basically gets delivered to the tRNA, and the tRNA has a triplet codon here, which is complementary to that in the mRNA. So if I draw in, for example, CGC, then the strand of mRNA, the triplet that's going to match that, I'll give it a go, see if you can figure out what it would be while I pick a colour for us to work with. It would be GCG, wouldn't it? There we go. And what this does is it basically picks out the appropriate amino acid. So this amino acid matches this sequence. And amino acids, so I'll write that in here. A acid, sorry, my writing's a bit wonky. Amino acids are the building blocks for proteins. So if I draw a little protein chain here, we've got heaps of little amino acids, which are our beads. And this is called a polypeptide chain. And polypeptide chains fold in specific ways according to what amino acids you have. So you might have an amino acid that's one pattern, one that's maybe coloured in, one with some dots. They're all different and they'll fold accordingly, so it'll make a funny shape like that. And that will be our protein. So basically this is the step that we're working towards. So you're probably exhausted from me explaining all of this to you. So for now, I'll leave it there. But perhaps if you would like to learn some more, you should join HD Academy and we'll continue our adventure through biology. Thank you.
That was great. Sorry about that a little bit in the middle. What I'll do is I'll just re-record the audio there.
Okay. Yeah.
So I don't think it's any much of an issue. How did you get to that stage? Did you like zoom out and stuff? Looks like you did.
Yeah, sort of like drew little boxes and then had little arrows and stuff. And sorry, I think my dad just called. Just see.
Yeah, he should be here by now. So.